what we will do now is move on to Dr. Ken Oye from MIT, Governance of Science and Technology. Ken?
Okay. First, that last slide that Todd had up is actually very close to my summary slide. And so I think that you know where this presentation is going to be heading. Let me back up just a little bit and note that what I'm going to try to do here is to use the charge to this committee as framing for the presentation. So if we look over, and I tried to reduce the words in the charge just a little bit, but let's kind of work through very quickly each of the things that you're being asked to do. And I have to tell you, I don't envy you the tasks that you have. Okay, so the first charge was to review the state of the science, of the gene drive research. And Kevin went through that already and did it very successfully. There's a timeline that we ought to keep in mind here. And the timeline is important because, man, are things moving quickly, all right? So you have nature doing a lot in the good old days because gene drives have existed in nature for a long time. Austin Burke comes along in 2003 with this idea of designed gene drives. Then, 2012, CRISPR-Cas9. I'm not taking a position on the IPR issue or fight over that. Notice I've included all of the labs. And then all of a sudden, 2014, Kevin, George, and colleagues make huge advances in terms of specifying how to go about doing it in practice, with lab research backing that up. Now, Kevin went over a whole bunch of those points that I have under 2014, but I want to note that in 2015, we have the UC San Diego group doing Drosophila. And the pace of change here is extraordinarily rapid. I also want to note a very encouraging thing. In 2015, today, embargo lifted 36 minutes ago, Science published the piece that Kevin and the other gene drive labs worked on together on codes of conduct to encourage responsibility in laboratory research. But I also want to note that that does not cover field release. And all of the issues, not all of the issues, 80% of the issues that your committee has to deal with go beyond lab safety and lab precautions to how do you want to make decisions on field release. But it's encouraging that there has been progress on lab. So I'm going to cut through these slides because Kevin has done it in much greater length, and turn to your second charge. So your committee was asked to use case studies on likely applications of gene drives to try to figure out oversight mechanisms that are suitable for three conditions: one, laboratory; two, release in advanced industrial countries; and third, looking to middle income and lower income countries. So let's move into that box just a little bit and talk about some potential applications. So one set of applications would be, well, okay, you can see here Todd looking fascinated as Kevin is talking with Dow AgroSciences and an NGO that's critical of gene drives. This was part of that exercise that Todd mentioned, where we had the example of gene drives as a prompt for figuring out what research needed to be done. Jim was a key part of that. We were kind of chairing some of those meetings together. And the applications that I think you should be thinking about as examples would include malaria, would include Asian carp or other species suppression strategies, perhaps zebra mussels. We begin with malaria, and we begin with malaria for good reason. We've had the Gates Foundation talking about the critical significance of it. We also note that malaria and dengue are endemic in areas that are not heavily represented when you look to where synthetic biology is being done. Okay? If we're going to be talking about governance mechanisms, let's begin with the observation that the areas of the world that are being beaten up by these diseases are not the areas where you have a huge amount of research taking place. This is the IGEM map that Todd put up. We can call this not incidence of malaria, but incidence of IGEM. And one of the suggestions I'm going to be making later on is that if you're really interested in having engagement, genuine participation, in those parts of the world that potentially benefit from the medical applications, that participation should also be informed by active participation in the research activities and programs, and we'll come back to that. But it's not just developing countries that are potential areas that you might want to be thinking about for applications. Sitting in the middle of that big blue spot on recorded cases of Lyme disease, if we're talking about attacking or reducing the ability of insect vectors to carry disease, as someone that returned from a camping trip to come to this meeting, I'd point to that as yet another potential application. Or you could be talking again about Asian carp, or even going after woolly adelgids and others in terms of invasive species. Now, observation going in, the beauty of gene drives, and we'll come to some of the potential risks in a second, is again, let's go back to the point that Kevin made. Genetic alterations that do not provide reproductive or fitness advantages for the species can be promoted through the inheritance biasing mechanisms that are at the heart of gene drives. And from the perspective of the malarial mosquito, the inability to carry malaria doesn't confer great benefit. From the perspective of the Asian carp being eradicated, methods of doing sex biasing are not necessarily great from its perspective, okay? And the power of the technology is the ability to drive genetic alterations through wild populations in a way that you can't dream about if you're sticking with old-fashioned Mendelian inheritance. And that's the basic point. But there are environmental issues and there are security issues that your committee is going to have to consider. So I'm going to actually try to take some of the points that Todd was making and some of the points that Kevin was making, but hammer on them just a little bit more with an emphasis on uncertainty. And here are some things that I think we need, potential areas of concern that we need to understand better. So if we're talking about environmental issues, gene drives are a little bit different from many other applications of biotechnology because gene drives, once introduced, will sit there and work their way through wild populations. And the mutation of gene drives is inevitable. We're not talking about one-time release, terminator genes, that kind of stuff. You're talking about releasing once with the organisms containing the gene drives modifying themselves over time. And we need to understand mutation and effects a lot better. Lateral gene transfer, we're not sure. Again, we need to understand that better, and Todd mentioned that as part of the research agenda, but we need to understand the effects of lateral gene transfer on the discrimination, the precision of the targeting that is actually one of the attractive features of gene drives. Okay, Kevin went over immunization and reversal drives, the partial undo button, and immunization as a means of limiting the spread of gene drives. Critically important because many of the precautions that you might want to recommend would be to get those in place before you do the primary applications. But we need to understand their effectiveness and stability over time. We need to make sure that the safety mechanisms work before you go ahead and do the experiments in the field. We also observe that diseases borne by vectors will evolve, and we also know that the environmental effects are not going to be one size fits all. We need to understand gene drives is not the issue. You have to understand specific alterations to specific species in specific environmental settings. And it's a hard and specific problem, a complicated problem, and not an easy one. Now, on to security. And if we talk about security issues, it's a little bit tricky. Kevin indicated that you're not staying up at night worrying about it all the time. You're worrying more about your son that kept you awake last night. But if we're talking about security, what I want to do here is push a little bit and come up with the most extreme statements that I can. Well, not quite the most extreme, but to push you guys just a little bit on it. So if we have the modification of insect vectors to reduce their ability to carry diseases, there would at least be the theoretical possibility of altering insect vectors to carry stuff. I'm not saying that any sensible, decent human being would want to do it, but it is a possibility. Or you could be talking about suppression of crops and livestock, not in modern commercial agriculture with controlled seed, but in traditional agriculture. a potential area of concern. If you can be talking about suppression of invasive species, potentially you could be talking about suppression of pollinators or other keystone species. And then there are the weird things. I'm a political scientist as well as a technologist. And you could be talking about immunization drives to protect yourself and allies from effects, which gives you something that's starting to approach a bioweapon. Or reversal drives might be withheld for economic or political gain. The point here is that bottom line. If we're really interested in understanding the security implications of gene drives a lot better, we don't know what they are right now. And speaking as someone who teaches at this crazy institution called MIT, my respect for the ingenuity of people to come up with stuff in hacker culture causes me to say you need to have people that are probing those security implications much more effectively. Okay, so let's go over very quickly a couple regs and then the concluding slides. Invasive species and suppression of invasive species. We have a little problem here. That would be regulated under a 2009 FDA guidance for regulation of genetically engineered animals. And you would have to demonstrate the safety and efficacy of the genetic alteration from the perspective of the Asian carp. I checked with this with Larissa Rudenko, who I would recommend that you bring in, and she confirms it, big problem. If you're talking about international environment, we've had discussion of Cartagena before, and we also have liability rules under Nagoya, Kuala Lumpur. The U.S. is not signatory. And I would recommend that we have extensive discussion of other mechanisms for governing effects because there's nothing in place right now. U.S. government DIRC policy. The DIRC policy does focus on a narrow scope of required oversight, and that narrow scope doesn't include stuff like gene drives. International security. It's broad when you look to the governing conventions. The Geneva Protocol looks narrow, bacteriological methods of warfare, but has been extended by analogy to viruses and would undoubtedly extend to malicious applications of gene drives. And the UN Biological Weapons Convention is also broadly defined, but scope of application is narrowly defined through Australia Group. So what I'd like to do is to move to some reactions and extensions and then summary. So the reactions to Kevin's work, to the science piece that we published, begins with a basic reaction, and not just from Friends of the Earth, from many. And it's really, oh my God, this is a little bit more interesting than traditional Roundup Ready seed. Then this reaction occurred actually in a room near here. And the idea is, what are you guys doing talking about this stuff? Don't tell the Muggles, leave this to the wizards. If you tell the Muggles, they're going to panic. And by the way, we shouldn't be talking about it this early because the work's not done. That argument weakened a lot when San Diego came in so quickly. For those of you who are fans of Harry Potter, this is a guy named David Gurwitz who published a letter in Science, and his position is that shut up, don't publish, don't talk about this, because it's so dangerous that any discussion in public is dangerous. Classify everything and have all discussion behind closed doors. The problem here is that you can't have honest discussions in classified domains of something with this range of power and with so many beneficial applications beyond. Now, Kevin and I made the mistake of doing a talk show. Those of you who have ever done talk shows realize that you get unfiltered reactions from the public. And Kelly from Millbury bugged me a lot. Her statement was, nature, malaria, don't mess around. And I have a daughter who was then living in Myanmar in an area where dengue and malaria is endemic. And I got mad. All right. The point being, though, that a lot of the public debate over these issues will not necessarily buy the assumptions that we're walking in with on the desirability of some of the applications. At the end of the day, gene drives do affect global commons, and we clearly need a global discussion of values and decision processes. And that discussion has to be informed and include technologically, scientifically competent and literate individuals. Takeaways. So the main takeaway that I'd like to offer is that right now we have some lead time. Not a lot, because the field is moving very rapidly. But look at this from the perspective of nerds, geeks, and wonks. And there are very important things that each of these groups should be doing now to take advantage of the lead time that has been provided. Nerds. It's really what uber nerd sitting over there on the other side of the room, Todd, was recommending. We need to better understand environmental and security effects. We need to get the research going so that the discussion on those effects and decisions on application are informed. But there are a lot of things we don't understand enough right now. Geeks, staring at Kevin, if you don't mind. Geeks, responsible geeks, wish to modify the organisms and the uses to minimize risk. And those strategies, those technical strategies, must be both developed and tested in advance. And finally, policy wonks. We sit in Washington. There are a lot of gaps in policy here, and those gaps have to be better understood. Red teaming needs to be done to understand the risks. The appeal for education to inform the technological community and public debate is well served. Resources, tons of stuff to read, but I really would recommend that you get into the room people like Celeste Schuma, sitting in the back, former executive director of the Convention on Biodiversity, Jenya Dana at State, working at the science office on these issues, and many others. This town, the country, and the world is rich in resources. The problems that you face are very complicated, and I wish you well on your very difficult endeavor.
Terrific.